that turbulence will cause shearing effect, and this mechanism of injury will start as well. So this is a very important point to remember. So anyways, so first damage that happened was reduction of the nitric oxide. Now what happens is, this LDL, which is oxidized LDL, is a favorite of macrophages. They love it, but there are some macrophages here, but not a lot. How do we bring macrophages here? Look, the oxidized LDL does three things. Number one, it is taken up by the macrophages, which are present here already. So let me make a big macrophage here. I should make that in the intima. Let me make a big macrophage here and put some LDLs in it, oxidized LDLs. This kind of a macrophage, which has eaten up lots of lipids, is called foam cell. It is big blobby foamy cell. And actually this cell would undergo a bunch of changes. I'll talk about that in a second. But the important thing that we want to see here is that this macrophage, this is one effect of LDL, that it helps create foam cells. Second effect of the LDL is it acts on the macrophages, one, it acts on the endothelial cell, two, and it acts on the smooth muscle cell, which are present in the tunica media down here. And it causes them to release cytokines. Presence of LDL is not invited, LDL is not needed here. So presence of the LDL, the cell starts releasing cytokines. What kind of cytokines? Cell starts calling inflammatory cells to say, please come save us from this LDL. So what are we going to see? We are going to see, so see here in the monocyte macrophage part of the process, what will happen is we'll have increased monocyte chemotactic protein 1. Where is it coming from? From the macrophage. So macrophage released MCP1. It also released interleukin 1. What will these two things do? They will bring inflammatory cells over. It would also do tumor necrosis factor. What would that do? That would cause, so I'm going to make one more endothelial cell. So see, now we have to go up. Why? Because the atherioma is developing. So as we go up, let's make a cell here. This cell, endothelial cell, now has on it vessel cell adhesion molecule 1, VCAM1. So essentially what is happening is LDL caused the cells, one, it got eaten up by the macrophage, second, it caused the cells to release cytokines. The cytokines have done two things. One, they are present here in the blood and the inflammatory cells will be pulled in, and second, it has caused an effect on the endothelial cell to catch the inflammatory cells. The cell adhesion molecules are extruded and made active and made exposed. So VCAMs are present now. The result of that is going to be what? The result is we are going to have more monocytes to come in, which will convert into macrophage. Now there is a third thing that LDL does before we go to other things. A third thing that LDL does, and that is it is a poison for the cells locally. Oxidized LDL causes death of smooth muscle cells, SMCs, it kills them, smooth muscle cells. It kills the endothelial cells and it kills the macrophages. So why is that important? This is tissue damage, guys, inflammation. As soon as the tissue damage starts, as soon as the cell debris becomes available, that in itself would trigger further inflammation and pull in the inflammatory cells, repair cells. So LDL's presence has caused three basic factors. Once that is all done, the process of inflammation has started. Now in the inflammation process, it's very simple. Monocyte is brought in because there is increased VCAM, there is tumor necrosis factor out there, there is MCP1 out there, there is interleukin 1 out there. Monocyte came in. Now can I say this, that at this stage in the atheroma development, there are some old macrophages sitting there fatty and fat with the LDLs. They are foam cells. Then there are some new monocytes coming in and they will become macrophage. Why am I saying this? The new cells that are going to come in, they're going to say, oh man, this is so much damage here, I want to work, give me work. This is the new cells that come in and start trying to bite stuff off that make the atheroma unstable. If the new cells were not coming in, if this current system just became stable by itself, it would be fine. It's the new cells that come in and say, oh, they start biting here and there. And as soon as they start biting, they make the whole thing unstable, it can rupture. Okay, so monocyte is in here. This monocyte will become converted to macrophage as well. And what would it do? Once again, it would release the reactive oxygen species. That is the function of the macrophage. I've done that in immunology. When the reactive oxygen species increase, what would happen? This whole cycle increases. Why? Reactive oxygen species works with the LDL to make it oxidized. You increase the oxygen there, reactive oxygen there, you increase oxidation of the LDL, you increase the process. See, that is why we say it is a progressive process, it won't stop. Okay, so reactive oxygen species increased. Interferon gamma, so before interferon gamma, let's say tumor necrosis factor alpha is increased. And TNF will do what? TNF will increase the activation. We are still in intima. Intima is now swelling inward. It would activate T cells. It will activate T cells. T cells in turn will activate macrophages. We have done this like 20,000 times in immunology. Interferon gamma will activate the macrophage, macrophage will activate the T cell. Now the macrophage is active, T cell is active, what will happen? There are going to be growth factors that are going to be released. The growth factors that will be released are very important for the final pathology or shape or maturity of atheroma. This is atheroma development. So what happens is the growth factors, platelet derived growth factor, where did they come from? They came from the damaged, so let me make red colored endothelial cells. So these endothelial cells are releasing platelet derived growth factors. The macrophages are releasing that. Even other cells like the smooth muscle cells are releasing it. Then there are fibroblast growth factors that are being released. Then there are transforming growth factor alpha from macrophages that are being released. In short, the platelets and the other cells present in the site of the atheroma increase the release of growth factors. Now that growth factor increase will cause the smooth muscle cells to start becoming proliferated. Now which smooth muscle cells? There are smooth muscles present in the media. There are smooth muscles present in the media as well, intima as well. And there are smooth muscle cells, their precursors that are running around in the blood too. Three sources: inside the blood, in the intima, in the media. The growth factors specially target the smooth muscles that are present in the lumen and in the intima. However, the media cells are also becoming target of that. Their phenotypes are different. The end result is smooth muscles start proliferating and start migrating from the media to intima. So they'll start coming here, and these smooth muscle would start going up towards the endothelium basement membrane and start lining up under the basement membrane. So this is the formation of a cap now.